at the modules, but please feel free to introduce yourself and also share a um, uh, surprising fact with us.
I can. Yes, thank you. So, thanks. Wow, it's been a bunch of great lectures. I'm just gonna start off with, introducing myself. I'm Fiona Brinkman. I'm a professor at Simon Fraser University. Has a long-standing interest in developing sort of more, quote, sustainable approaches for infectious disease control, and as a computer geek on the side. So I got into increasingly my wet lab as more just computational work now. Um, I'm coming to you from and I will bring up my fun fact in a second. I'm coming to you from the Coast Salish People's Lands, Tsleil-Waututh, Coquitlam, Squamish, Musqueam nations. They're really beautiful lands that I live, work, and play. And I would argue I also, oh, wait, what's going on here? There we go. I also go for rides in various places on these lands. And my fun fact is I like to come up with any excuse to celebrate stuff. So I look at the fun time and dates, fun holidays, and we just come up with excuses to do things like um, like go for a ride day today is, you can go and hop on a bike and go for a ride. But yeah, I want to also just sort of start with a little bit of a big picture. So normally this lecture is near the beginning of the workshop. I was a bit worried that, it's bringing up a lot of basics. So I thought, oh, should it be at the end? But actually, I realized maybe this can be good, is it will ground you. And I really encourage you guys, have been through a lot, just to really sit back, relax, think about the basics of all of this and make sure you're sort of comfortable with some of these basics, including understanding fundamentals of like character-based evolutionary analyses, phylogenetic analyses, learning how to simply interpret a tree, including using it to predict function, knowing the basics of how to build trees, and appreciating different kinds of components of this, including phylogenetic bootstrapping and the benefit of looking at unique characters in convergent evolution, which sort of touches on some points that have been mentioned throughout. And so hopefully, some of these basics I can sort of refer back to some components of the lecture and it'll be useful. But I would really love it. This is the last module, and I would love it if you can bring up any comments later of what we will have a survey, but just hearing what you like and don't like, and whether it's actually useful to have this as sort of like a last session. Okay, and I'm going to keep on, just a minute. Yeah, I'll try to keep on my the Slack so I can sort of see if there's any questions there, but please don't hesitate to just speak up, anyone, if you see something. Okay, so first, just a reminder that microbial genomics is important, and that contextual data associated with microbial genomics is important. So, um, we talk about genomes, but they're really not very as useful unless you have that additional sort of metadata or contextual data associated with it. This comes up in the next workshop that follows after this more. But just a cautionary note too that large language models and generative AI, just before I forget, have been transformative. They looking at the language of DNA and proteins as being really becoming very powerful. But just remember that the nature of these models is they are forced to predict the next word. So they can easily, because of that being forced to make predictions, can start to make errors and you can get what they call hallucinations. And particularly when you're dealing with edge cases of, say, curating data around sort of trees or doing analyses of microbial genomes, you can get these problems. So I liked Russ Altman's description. It's like a cocky third year medical student will very confidently tell you something that's not right, particularly regarding edge cases. And here's a picture of when I asked ChatGPT to give me a picture of a cocky third-year medical student. This is what it gave. So this is your ChatGPT right now. But that said, there's very powerful ways it's being used, and I'd be happy to talk about that more at the end. Um, but let's step back first. Let's just talk about just what this is all about. We're looking at these molecules as sort of documents of evolutionary history. So how did this theory first evolve? I want you to appreciate that, this is a theory that did evolve in the context of a lot going on. So there was an appreciation that the world was not constant, but changing. Plate tectonics were being developed. Discovery of fossils were accumulating. And then they were wondering, like, are these remains of unknown but still living species that are elsewhere on the planet? But Cuvier had the really landmark study showing that the deeper the strata, or you dig into the earth, the less similar fossils were to existing species. And then you'd see fossils that would match with some of the plate tectonic theories of the coast of South America and Africa having been joined previously. So it's pretty cool that all of this stuff was coming on together. And in that context was Darwin's Origin of the Species, the origin of species. And really what was landmark there was coming up with a sort of an approach and an explanation for like a mechanism, I guess, for how to think of organisms and that all organisms are derived from common ancestors by a period of a process of branching. And this really explained the fossil record, explained the similarity of organisms classified together, including families, shared traits inherited from a common ancestor, and the similar species in a similar geographic region. And really what's come down to it is nothing in biology sort of makes sense except in the light of evolution. So this is really taken off as a way to look at things of having a tree where you might have sort of your living species, but always remember what you're looking at often in terms of living species, just a snapshot in time, right? But there's a whole sort of process that's occurred before that, including some, you fossil species, for example. But so evolution generally I think of it as descent with modifications, changes in heritable characteristics of biological populations. So not if you get the sequence change on your skin, it's not going to make a difference. It has to be in your sort of germline. And in bacteria, of course, the whole bacteria is basically the inheritable as the inheritable characteristic. But you have different kind of processes going on. There's natural selection. You'll have heard of survival of the fittest and that kind of selective evolution. But just to want to remind you, there's also the sort of neutral evolution. There's just we call it a molecular clock of ticking sort of changes occurring in nucleotide sequence or sequence over time. The biology is beautiful and that it has these errors that are occurring sort of built in that provide this diversity and leads to being able to survive, when there's something will survive because of this diversity when you have some sort of environmental change, for example. So these evolutionary processes give rise to biodiversity, estimated over 8 million living species. Basically, not cat. We don't know all of them yet. And one thing particularly relevant to this work is I want you to appreciate that, microbial evolutionary processes really blur the boundary of species, as we sort of started to allude to. There's asexual reproduction or clonal kind of fission of bacteria, but there's also, as has been mentioned repeatedly, horizontal gene transfer, such as infection by phages, for example, though like there's cool bacteria like Neisseria. I used to study, have this sequence GAA, GTC, TGAA, that basically are oh gosh, I've forgotten the sequence. That's horrible. I'll have to remind myself about that. But the sequence is basically in the DNA and it allows these naturally competent bacteria to uptake DNA from the environment that has that same sequence in it and allows it to do exchange as well. Just one thing before we start talking about sort of more phylogenetics, I want you to appreciate that to study history, one must know in advance that one is attempting something fundamentally impossible, yet necessary and highly important. So always remember that when we're doing these analyses, it's allowing us to estimate or infer evolutionary relationships. We have seen some evolution, though I would argue um, in practice, we see evolution of, and within 10 years of any antimicrobial being developed, we start to see resistance to that drug showing up more in certain environments. We've seen, there's the famous Manchester moths that were like, um, you know, whitish and then changed to dark color to better camouflage in the sooty Manchester Industrial Revolution. So there's been things that we've actually documented and seen occur. But but just remember that we are generally trying to infer things, and but it can be very powerful to do this. So first, before we get into that, some terminology. So we've got systematics is basically the study of the interrelationships of living things. Taxonomy, I always like to point out, is the science of naming, classifying organisms. Evolutionary theory is not necessarily involved in taxonomy. It's just that classification. However, increasingly, we are using the power of genomics and these sequence data as a way to classify things that are using evolutionary theory more. So you'll see a lot of organism name changes, etc., that will occur as people try to do this. Um, Phylogenetics is the field of systematics that focuses on evolutionary relationships. So that's really when you're looking at the phylogeny or the evolutionary relationships between organisms or genes or proteins. You can look at it from a few different perspectives. So um, phylogenetics involves an analysis of a set of characters traditionally were morphological features. You could look at the morphology just to be a bit Canadian for this Canadian workshop. We have deer around, and I'm not to say that there aren't deer elsewhere, but we have moose, traditionally are noted, and look at those great moose antlers. But you could look at these morphology and these features of these antlers and look at the evolution of this kind of feature. But now, what we can do is combine morphological and molecular sequence analyses and really get a really nice evolutionary tree of how these things are evolving. And what's really powerful is that each nucleotide or amino acid in your sequence is like a character. So you're able to look at thousands of characters at once, and that's much more powerful in terms of identifying things. I'll come back to things like looking at unique characters or convergent evolution. But I do want you to appreciate that these methods are depicted usually as phylogenetic trees with several assumptions. So there's usually an assumption of a bifurcating pattern, a pathogen. That was a Freudian whip. But bifurcating pattern of divergence, and that sequences are homologous. They have to have some sort of shared ancestry. So we're sort of saying, so if you have horizontal gene transfer, I'll come back to that later, that really mucks up doing phylogenetic analysis. You're assuming that everything is homologous and has shared ancestry, and you have this bifurcating pattern. But of course, that bifurcation can occur like right away, so you end up with like a bunch of things going to branching out around the same time. Um, but let's just go over the tree terminology, and this is where I think maybe this would be helpful to get earlier, but I think hopefully it's just useful for you to sit back and look at this. So remember that phylogenetic trees consist of nodes connected by branches. The terminal nodes are called leaves. We've also have called them operational taxonomic units in some cases. They represent the sequences or species where the data was obtained, and they usually represent living species. The internal nodes represent hypothetical ancestral species before a divergence event. And then the ancestor of all the sequences or species in a given tree is called the root. Now, not all of them have a root, as I'll bring up, but usually you have clades. You're looking at not just two sequences. That's obviously not very useful for making a tree. But if you have a clade, you can look at multiple sequences, and you will have usually a clade, which is a monophyletic group that includes a group of species that includes the most recent common ancestor of its members and the descendants from that clade. So, for example, sequences A and B are, and this is a clade, sorry, with sister taxa that are basically species arising from that same node. So A and B here are sister taxa, C and D are sister taxa, and clade 1 and clade 2 are sister taxa. Okay. And feel free to interject if you have any questions at this point. So the tree types, you have a cladogram that only shows the branching order. The branches don't have meaning. They're just showing the order. In this tree, for example, a tree for 3 sequences are shown A, B, and C. This tree indicates that A and B share a common ancestor more recently than either does with C, assuming this is a root here. But I want you to note that the different shapes can be used to show the same thing. So this is the same, exact same tree as this tree, but it's just a way to show it as a nice, vertical lines. If you have stuff that you want to mention, like whole organism names, it's sort of easier to put them here, and you just have some spacing to separate them. And so I guess what I should mention is these vertical bars are here, just separating them. There's not really meaning anything. So note that you could have phylograms. This is where you have branching order and scaled branches to indicate some attribute of similarity, such as the number of sequence changes. So this tree indicates that A has acquired more substitutions than B since they diverged and from a common ancestor. And the branch lengths basically can be indicated using a number or by adding a scale bar. You often see a 0.1 to indicate number of substitutions per site. And the distance, though, here is always that vertical line. So here it's saying the distance between A and B is 6, and the horizontal branch lengths are not meaningful. They're just used again to separate the taxa. The distance between B and C would be 1 plus 1 plus 4, right? For example. So again, when drawn vertically, the distance between any two nodes is the sum of these vertical branches. When drawn horizontally, the distance between nodes is the sum of the horizontal branches, if you want to draw it the other way. So again, you could have this tree orientation for a phylogram drawn vertically or basically horizontally, where here it's the horizontal branches that count in terms of distance. So also the other thing I think is always important to point out is a reminder that the order of the leaves here, that you can rotate around them. Think of it as like a mobile that you would have above a kid's a crib or something, how they can rotate around. So this is like a mobile, but rotating the branches doesn't change the relationships, okay? It's not that somehow A is more closely related to D here because it's like really close here. What counts is that D is close to C here because of that branching order. And you can just rotate these things around to show different things. This is all exactly the same tree. The relatedness is depicted by the tree topology of how things are branching. Note the benefits of unscaled versus scaled branches. So you can have unscaled branches shown here for some E. coli VTech phylogenetic analysis. Or if you scale it though, you get to really see that, oh, these two are really quite different from all of these guys. But these guys are really similar, so it's actually really hard to see the tree here. So what you can do sometimes is you can come up with different ways to show it. You can maybe have a scale And you might say like a little dot dot, just sort of showing that there's like these little guys, but they're really closely similar here, versus this guy that's really divergent. So there's different ways you could scale with visualization of the clades. You can just scale it in general, or you can have sort of unscaled, and they can each be useful. So then I want to talk a little bit about rooted trees. And rooted trees is where a root is basically the ancestor of all the sequences in the tree, where you have sort of like a descent, where this is sort of like older and this is newer in this particular example, right? The tree with the root shows this order of descent, in terms of implies A branched off earlier and C and D um, diverged more recently. Um, Unrooted trees are still very popular. This is a really bad version, actually. It should be sort of like branched out a little bit differently there, um, but an unrooted tree basically lacks a root. It doesn't show the direction of evolution. It's less informative. It's often drawn in kind of a radial format, though this is not the best radial format. But main point I want to point out is that roots are really helpful because they can really show the relationship. If you don't have a root, you don't know, is this the base? And then human diverged first or whatever. So what you do is you often use an outgroup or a sequence that's thought to be ancestral or more distantly related to any of the in-group members than they are to each other. For example, to root some mammalian sequences, you might use sequence from a non-mammalian vertebrate as an outgroup. So you've got these mammals, and you could use a zebrafish and put a zebrafish sequence in here. And then you can see that the zebrafish is there, and then you can see that cow diverged first, and human and chimp diverged from each other more recently, as certainly we're familiar with. But if you didn't know, if you're looking at a bunch of bacteria, for example, having that root can be really useful to see that order of evolution. So here's an example with uh, bacteria, where if you're looking at a bunch of E. coli, you could add a root, and you could look at add a root for salmonella, and you could see, for example, that 0157 diverged sooner than your classic E. coli K12 used in the lab lab as a lab-based non-pathogenic strain, versus it is more closely related to 0104, for example. Or I just had to, because there was uropathogenic people studying um, um, I guess uh, looking at caring about urology, I thought I'd throw in some UPEC. Here's a similar kind of relationship with UPEC is a little bit more uropathogenic. E. coli is a little more divergent than E. coli or these Shiga type Shiga E. coli, for example, interpathogenic E. coli. Um, so why root a tree though? Like again, without is human more similar to gorilla or chimpanzee? Without a root, you can't tell from this. But if you did a root without group, oh, that's a bit blurry. You can see that actually gorilla diverged first and human and chimp are more related. So this can be very useful for just getting a sense of what came more recently. Okay, so let's just take a second though. And by the way, I meant to mention we'll talk for a bit over an hour, and then we'll have a break for 30 minutes, and then we'll do a little bit more lecture in the context of an interactive session. So there isn't going to be any kind of lab where you're working at the command line or anything. This is just going to be an interactive Slido-based session after the break. But I do want to still give you a second here. Just take a second and look at this tree, and feel free to put in the Slack, what does this tree tell you? Does this tree tell you anything about relationships between bovine, human, mouse, or just in general, what does this tree tell you in terms of what kind of tree it might be or anything like that? Feel free to just stick in the Slack just any kind of information of what you might think this tree might be telling you. And just note the A, B, C, and D are just to sort of talk about, be able to refer to different branches in the tree. Again, remembering that the horizontal branches are more, they're more, they tell you something about the tree, whereas the vertical branches don't really tell you anything. So I'm looking, I'm not seeing anything in this Slack. I don't know if oh, apparently we're more related to mice than bovines. Correct. That is actually true. In some ways, okay, and I'll explain why. Okay. Branch links, the things that I'll tell you about what this tree does tell you. The branch links differ, so likely it's a phylogram. Um, it appears to be rooted because there's a line here, but you need to check the methods to confirm. Um, Human and mouse do seem to share a common ancestor, right? But the mouse lineage has really undergone accelerated evolution. And because of that accelerated evolution, it has a much longer branch length. Humans actually, if you do look at a, a sort of a distribution of our similar sequences between human and mice and human and bovine, we actually do share more sequence similarity with cows than mice. This tree is drawn, though, a bit with a little bit of exaggerated branch lengths, and it's sort of more overlapping distributions of similarity. Yeah, but yeah, so these guys really did evolve more rapidly. Um, they do have shorter generation time, which is one factor. Okay, so let's do another example here. Take a look at this tree and feel free to put in the Slack another example of which node do you think represents the probable ancestor of North American bears? So you can see there's North American bears up here, Asian bears, and I picked the bear example just again to be sort of there's bears around here. I'm in bear country in Canada here, but yeah, so just feel free to put in the Slack what which node, A, B, C, D, or E, like just put in what letter do you think represents the possible probable ancestor of North American bears? So which node would you think it would be? So I'm seeing C show up, and you are correct. Exactly. Yeah. Sorry, I should have let everybody respond. Apologies, but with the Slido, we'll be able to do that better. In terms of C is the node that represents the probable ancestor of North American bears. If you wanted to look at the ancestors of the polar bear and brown bear, that would be, for example, node E. Just for the record, black bears are pretty nice. Brown bears and polar bears, not so nice. Don't want to be near those. Grizzly bears, definitely want to keep away from those big time. Anyways, okay, so fun fact, just as a little aside, because of I care about microbes. The panda's ability, pandas are even more distantly related to these bears that we were looking at. Panda's ability to sustain itself on bamboo diet seems to be more dependent on its gut microbiome that has key involved in protein metabolism, including key amino acid synthesis as a major energy source for pandas. So, yay for we could argue that our like what we are as an organism is really not just one organism. There's a lot of philosophy there about really what is an organism now. We do need our gut bacteria are very important in multiple ways. Okay, so but let's go and take a second. Oh yes, Rob made a comment regarding bears. If it's brown, lay down. If it's black, fight back. If it's white, good night. I remember that. Thank you for reminding me of that. Yeah, so how do we build a tree? Okay, so I'm going to go through a few sort of lightly touching. I'm not going to be going into it in detail, but I want you to appreciate when you're building a phylogenetic tree, there's really three things you need. You need a multiple sequence alignment, a model of evolution, and a tree building algorithm. And the objective is to identify a tree that best describes the data given the model of evolution used, okay? I can't emphasize this enough. The multiple sequence alignment part is pretty important. You know, the sole purpose of multiple sequence alignments is really to place homologous or shared ancestry positions of homologous sequences into the same column. So when you do a tree, make a multiple sequence alignment based on a tree, you're saying that this is all homologous, and you might want to think about it. You know, what am I thinking here? Is this glycine, should this be over here because there's other glycines here or not? You know, what am I saying is my columns of sequence? Because that is what you're trying to say, and you're trying to use those as characters, just like you might use characters of antlers, for example. Um, and then, so you're saying that each line column is homologous. You're using a model of evolution. I'll talk about this more in a second. Transitions, transversions, for example, and how much they occur might be a model of evolution for, and then you have a tree building algorithm, how you're clustering the sequences. So I'll come back to that. So there's 4 steps again. Or actually, yeah, take that back. I lied before. 4 steps. So there's constructing the sequence alignment, evolute the model, building the tree, and then evaluating the tree is also really important. And I can't emphasize enough, contextual data and sequence quality are critical. Good quality sequences labeled with relevant contextual data will make a huge difference. If you have a non-homologous sequence that you stick in there, it can make your tree very messy. Um, so there's 2 main tree building methods I'm going to go over. Distance-based, where you transform the sequence data into sort of pairwise distances, and character-based, where you use the aligned sequence directly during tree building. So the distance-based methods, we create a tree based on a distance matrix and built using a multiple sequence alignment. The simplest measure of distance between 2 sequences, just literally the count the number of sites at which they differ. So for example, here for this alignment of sequences A, B, C, and D, you would look at, for example, for A and B, you might look at how many changes they have. So I'm looking at A and B, and I see that there's 1, 2, 3 changes, and then these spots are all the same. Um, whereas, you know, A and A itself, of course, have no changes, and C and D, for example, have 2 changes here versus this. So you might sort of say that there's distance, these are distances between these sequences. That's a very simplistic example. Obviously, we want to weight the nature of those types of changes. So we tend to use things like blossom matrices, other matrices that I can go into if you're interested. But you know, but appreciate that there's different kinds of methods though to look at them, and then we use neighbor joining, for example, for doing the tree. So here's an example of you know, doing a distance matrix method. What I want you to point out is it's easy to go from a tree to a matrix, but a tree that exactly fits the matrix often doesn't exist. Distances must satisfy a lot of mathematical properties. So we want to find the tree that most closely matches this matrix, this distance matrix. It turns out to be what we call an NP-complete problem, and we need heuristic or approximate approaches basically to work this out. So neighbor joining is a heuristic method where at each step joins the 2 closest subtrees that are not already joined. It's basically you start out with a completely unresolved tree. You take all your sequences and you go, okay, I'm going to put them all as a sort of star phylogeny, A, B, C, D, and E, and F. And then you do a matrix from the original distance matrix to determine the average distance from each node to all other nodes. And I'm not going to go through, I'm going to go breeze through this a bit, but just say that based on the distances of you basically can come up with the A and B are most similar to each other, so those two should be joined. And then you come up with a new node representing the fact that you're joining those two. And this is what we call star decomposition. You're going from a star to a phylogeny and you're joining those two. And then you look at the relationship of those two versus C, D, E, and F as a sort of new node. And you end up basically with a tree that basically looks at A and B together first, and then C, and then F, and then E and D, for example, in this particular example. In short, the neighbor joining tree, though, to cut to the chase, it provides an unrooted tree with branch lengths. The tree can be rooted if one of the sequences is known to be an outgroup. Neighbor joining, though, won't necessarily find the optimal tree, plus it only ever outputs one tree. There's other methods like parsimony that can find more than one tree, the most parsimonious explanation. However, extensive testing has shown that this method works pretty well to give you a general sense, and it's relatively fast. So, one of the things I'll talk about later is, what method to use for what. But if you're basically just trying to get just a general sense of relationships, neighbor joining is pretty good. But certainly a lot of the time people are interested in using character-based methods, also called discrete methods, that operate on sequences rather than just putting everything down into distances. If you take a sequence and you collapse it down a distance, you lose a lot of information about that, including like where gaps are and natures of things that exist there. So two major character methods are maximum parsimony, as I sort of alluded to, which finds the most parsimonious method, and maximum likelihood. Maximum parsimony is finding the tree that is the fewest evolutionary steps. Again, it can find multiple most parsimonious trees, which is sort of cool. Maximum likelihood is, I'm just going to go into a bit more, involves finding the tree that's most likely to produce the data, given a model of a sequence evolution. It's very popular because, like all character methods, it uses all the data and it gives a probability for your tree, which is really nice. So maximum likelihood, again, it tries to find, it maximizes the probability of observing the data given that multiple sequence. Oh, I'm seeing a comment here. Do we need to have a cutoff to consider anything below that within a cluster or similar? No, so the good question. So you basically you're sort of coming up with your sort of what you're saying is most similar based on your model of evolution. And then it's building a tree reflecting that model of evolution and the similarity of the sequences. Okay. But yes, there are multiple different kinds of approaches for how you can cluster things. Certainly be happy to talk about that more at the end, I guess. But what I want you to appreciate here, getting back to this, is that this requires a model of sequence evolution, a tree, and the observed data. And so otherwise, given data D and a model M, find tree T, given the probability of, or the probability of D, given, that's what the vertical bar is, given tree T and model M. And so we try and make this probability is maximized. So this requires examining all the columns of the sequence and searching through all the possible trees. So it's more computationally intensive. So I'm going to walk through this a little bit just to give you a sense of that very, very generally. Okay, it's very light look at this. So first, just remember evolutionary model. I sort of alluded to this, a classic example of an evolutionary model is weighting more. If you're doing nucleotide sequence, weighting more transitions versus transversions. You guys should be familiar with the fact that transitions going between G and A or going between C and T are sort of certainly more common and easier to have. I sort of think of it as ag and cut. But transversions are a more significant change and less favored. And So transitions are generally occur at higher frequency than transversions. So when you see a transition, you might want to weight that change differently from a transversion, which is sort of more significant. Um, here's an example, though. Let's walk through an example of consider these four sequences and this tree, 1, 2, 3, 4, right? So you basically saying 1 and 2 are together and 3 and 4 are together in this example tree. So you want to look at what is the probability of this data at position 1 only, given this tree only, and a model of evolution that says the transition is 0.3 and transversion is 0.1 and probability of no change might be like 0.6. So we're going to go through this and we're going to calculate the probability by putting this A, G, and G here, on the tree. And uh, we're going to calculate the probability of the tree for every possible reconstruction of ancestral. I'm just realizing my watch is beeping at me. I should stop that. Okay. Uh, we calculate the probability of this tree for every possible construction of ancestral sites of GATC at position X, so this sort of ancestral node, or position Y. So there's 16 values to calculate, 4 times 4 of GATC, right? And so we do this, we look at this 16 values, we can determine the probability of this column given this tree and this particular model of transitions and transversions, okay? And we're doing that, okay, just for that one column and one tree. But other positions are calculated, all the different columns of the sequence, and we can do that for all the different columns of the sequence. And we also can do this for all of these different possible trees. So that's the likelihood of one particular tree, but we need to also look at it for looking at these different possibilities. Here's 1 and 2 together, here's 1 and 3 together, here's 1 and 4 together. Okay. So in short, what it means is it requires searching through many possible trees. It's computationally intensive. But still really good because really looking at all of the data and it gives you a probability. I'll just add a couple of notes here that the evolutionary model can also include time. This means considering each topology and best branch lengths for each topology. There is this whole other course where we go into phylogenetics, the other workshop after this. But I do want to really emphasize the result of this maximum likelihood is dependent on the model of evolution used. So you've got to sort of be careful to look at your model of evolution, see if it's suitable. So there's other approaches too. Bayesian approaches, look at Bayes' theorem. It has the ability to incorporate prior information for events like prior distribution for outbreak onset time. So I do want to emphasize this is a really popular method too for looking at when you're looking at sort of, for example, outbreak analysis. I also wanted to really comment here that recombination invalidates most approaches since your columns of your sequence should be homologous or have shared ancestry. So it's important to detect possible recombination before performing phylogenetic analysis. And I guess I see there's some people doing comments. Oh, thank you, Rob. It's great providing some more comments. Thank you. That's a really good point. For ultra-fast trees, one thing that got developed as a result of, this really the pandemic has benefited from was we had all these SARS-CoV-2 sequences, insane numbers of sequences. But so coming up with Usher as a method for ultra-fast placement on existing trees was good. That enabled placing a sample on a very large tree and not having to recompute the whole tree again when you would do it. So just consider this like a taste of different kinds of methods that, honestly, you can take whole courses in learning about these and encourage you to get familiar with them. So what is the best tree building approach? So there's no single best method for all circumstances. It depends on what you're doing. You might want to use like the Usher kind of approach if you're just sort of trying to quickly find out where a sequence might be placed on a tree. If you've got time and res computing resources, you might want to perform a more computationally intensive classical machine learn maximum likelihood method. Um, if you're trying to identify a model, there's different kinds of, you can try a couple and see how they fit the data. J-model test is an example. And I just want to note that I was going through my slides and noticed this link is no longer working. It's all in GitHub. So I updated that link. And oh, before I forget, I've added in extra slides, and also there's the slide results from the Slido. I'm going to post in the Slack a copy of these slides for reference for you guys, the sort of after version of the slides that contain a little bit of extra material, including some answers to things. So just note that. Yeah, I'm just trying to see what was written here. There's more of a, oh, that's so awesome, Rob. Thank you. He's bringing up more about trees. Okay. So another consideration, I just want to remind you that we can sort of look at proteins or we can look at nucleotide sequence. Um, RNA, DNA, you have more informative sites when you're looking at a protein coding gene. You've got three um, bases for every codon. For very highly related sequences, that can be useful. If you've got protein sequences that are basically identical, I mean, you can't really do much of an analysis with that, but you can see sometimes that molecular clock like ticking third base codon change. But for more divergent analyses, protein sequence often wins more divergent characters. There's more different characters, I mean, and so it's better for more looking at more distantly related sequences because a G could change to a C and back to a G very easily, whereas protein sequence, there's more amino acids. And there's also much less bias. You don't have like the codon usage bias that can occur that was also alluded to in some of the chat as well. For aiding tree evolution, you've got bootstrapping that also is a powerful method to sort of test whether the whole data set is supporting the tree or if the tree is just a slight winner among equally and nearly equal alternatives. So it involves you just randomly perturb the data set, usually 100 to 1,000 times, such as, adding in an extra column, duplicating a column, that kind of thing. So here's the original data. You might have a bootstrap replicate where this is all the same, but you've changed this to a TAC, for example, here. Sorry, no, wait a minute. No, you've changed here. The bootstrap replicate is you've duplicated this column. Here, the bootstrap replicate is you have a duplicate of these two columns. And here, the bootstrap replicate is you have a duplicate of these two columns, I guess. And what I want you to note is it'll show you you do the original data, which shows B and C cluster together more than A. Bootstrap replicate 1 shows the same kind of thing and shows the same kind of thing, but the bootstrap replicate 3 actually, wait a minute, shows A and B going together. So what you do is you say, okay, 66% of the time, or 2/3 of the time, this kind of structure was seen. So I'm going to make a final tree showing that B and C most likely they came together with bootstrap support of 66% of the time. Now, 66% of the time, something going together doesn't show sort of strong, necessarily, I would argue, strong support for the tree. But the main point is then you perturb it. The tree is built using the same method as the original tree, and the original tree is labeled with these at nodes with the number of times out of whatever. In this case, it might be 100 out of 66 out of 100 times. You might have this consensus. Now, a bootstrap value, I would argue over 70% is considered good support. I would argue you want higher than that. But one thing I would like to emphasize is that due to this random perturbation, each time you bootstrap, you can get different branching orders for branches with lower support. So it's not like you make a tree, um, you're going to get exactly the same result each time because of that randomness. But here, let's look at a tree. This is all the same text here, but let's look at a tree and see what we get. I'm just checking if there's any comments. Yeah, so here's a tree where, 100% of the time it's saying out of 100 bootstrap value, we're saying these two diverged first. And then if this is assuming this is a root, but you don't need to confirm that that's actually been rooted. 100% of the time, these guys and these guys are sort of diverging as two different clades. But what I want you to note is here you've got some lower bootstrap values. So it might be, for example, that sometimes this ED1A and 536 might cluster together, for example. So it tells you there's some uncertainty there, right, with regard to what you have. And then I want you to also note that just high bootstrap values provide support as long as the multiple sequence alignment is robust. If you have a really bad alignment of one sequence, it'll give you 100% of the time this out in left field branch, that's for that. But that's just because that sequence is misaligned or not even homologous in some cases. So you do need to have a good multiple sequence alignment. I just thought I'd throw in because some people are doing soil bacteria, and I care about Pseudomonas, which are found frequently in soil and are noted for their intrinsic antimicrobial resistance. It was mentioned, the Pseudomonas were mentioned earlier. They actually have a very intrinsic lower outer membrane permeability that they're basically found on your fruits and vegetables. They're all over the place. But they're pretty noted for impacting certain people are susceptible, and burn victims also are particularly susceptible. It's a nasty bug if you get it because it's really hard to treat. Immunocompromised people, those who are in hospitals and who are particularly vulnerable. But main point I want you to point out is that here you can see this, Pseudomonas and these different kinds of Pseudomonas species, some which are sort of protective plant promoting, growth promoting bacteria, and some which are plant pathogens, and some which are human pathogens. Um, I want you to appreciate that, you get all these 100 bootstrap values, which is great. You can see these different groups. But just remember, because you're rooting with E. coli here, it pushes all these guys really close together, and you can't really see that Pseudomonas aeruginosa, for example, has really multiple different kinds of major groups. And so just be aware that you have to choose your outgroup also carefully. If you do something too divergent, you're sort of you're going to lose some information here about the relationships. So you might want to sort of use E. coli as your outgroup to get a sense of the general trend of these guys. But maybe if you're looking at Pseudomonas aeruginosa, you might want to use another Pseudomonas species as your that's more closely related as your sort of outgroup to basically get a sort of a more refined look at that particular clade. Okay, so some fun stuff to me anyways. I always love looking at these cases of unique characters and conversion evolution. So I just want to remind you, we can look at these whole sequences, but just don't forget to look at those interesting things that show up that are somewhat unique. So unique characters are basically things that occurred once and are unreversed. Example is fur. If you go into the middle of the Amazon and you find an animal with fur, that is very likely a mammal. And if you see something with feathers, that's very likely a bird. So they're very useful for inferring membership in a group and inferring relationships. A genomic epidemiology example, where you're looking at microbes, you might find an indel only in sequences associated with outbreak in a certain geographic region. Phylogeny suggests a new sequence of interest elsewhere that might have originated from that location. And if you're seeing that sort of unique indel, maybe that would further support that phylogeny and that hypothesis that that sequence originated from that other location. That said, you have to watch out for the fact that things can occur. Oh, here's another unique character. I just thought I'd mention this in light of the talking about species that here's another microbial genomics example that I found really fun. We found plant-like genes in chlamydia species, which really turned out to reflect that chlamydia have a very ancient ancestral relationship with cyanobacteria and the chloroplast. So basically, if you look at, and then to sort of support that, we looked at unique ancient characters and we looked at the ribosomal superoperon, where there was this shared loss of this S10 and S14 in Synechocystis, which is a cyanobacteria, and chloroplast and chlamydia that sort of really link these. These are found elsewhere in the genome, but this loss in the superoperon was able to sort of help provide more support for the fact that these guys likely have a very ancient evolutionary relationship. So just as a reminder, this is the endosymbiotic theory. Chloroplasts are thought to be sort of like ancient, have an ancient relationship with cyanobacteria. And basically a cyanobacterial ancestor, went into a eukaryotic cell, and then some of those genes from that ancestor, this microbe that literally went into this ancient eukaryotic cell, they moved into the eukaryotic genome and then are targeted back to the cell of these microbial cell or chloroplast ancestor into the chloroplast, and that sort of links them to basically there's so you look at a microbial uh, uh, if you look at a eukaryotic genome, you will find a bunch of sort of things that look like they're bacterial, but they're actually related to the mitochondria, or you'll find things are related to, in plant cells, you'll find these plant-like genes, but they're just because they're genes that are ancestrally related to the ancestor of the chloroplast that are targeted, these proteins are targeted back. I probably could explain that better. Sorry, I can find a link to explain that. Main point is that you have these unique characters, but I also want to emphasize this convergent evolution that occurs too, which is cool. My favorite example has got to be all these animals that had legs that went back into the water. So, for example, um, it's an ancestor of an elephant that went in back into the water, and those legs basically fused and became oh shoot, I'm blanking now. What do you call them? Not fins, flukes. So they formed flukes. And so they basically their legs fused to form these fluke. An ancestor of a canine dog went back into the water and formed a walrus, and ancestor of a sort of one of these grass-eating organisms, a cow, or an ancestor of, pigs and alpacas and cows and stuff, basically formed led to the formation of dolphins and killer whales. So what's notable about this is that they all formed the same kind of fluke kind of thing because it was advantageous, of course, to stick those legs together, and they all go up and down because of that nature of their original ancestry. Similarly, you can look at mutations, for example, strong selective pressure to enable mutations leading to increased antimicrobial resistance. For example, you can see this occur multiple times independently in different lineages. And so, in short, when you see a particular mutation occurring frequently, independently in different lineages, take notice because it can indicate a functional advantage. I'm actually going to hit you with one more because we were talking about the idea of species and whether they can be have similar functions and then sort of converge and be more similar in sequence. Here's another cool example. Antifreeze proteins, basically, they all have a threonine-rich flat surface that water molecules spaced like ice will bind to that surface well on this light-colored area. And then there's this opposite surface, which is all disordered. And the idea is that if you have ice forming in an organism, that's bad. It can poke holes in your membranes. So like these organisms that live like snow fleas live in very cold environments have these ice sequestering proteins that basically bind ice. They have a disordered sequence on the other side to help the water stay as water and not form an irregular structure like ice. And these help these organisms live in these very cold environments. But what I want to point out is the sequence of these are not the same. They've evolved the same kind of feature, but the sequence, these evolve separately and have no sequence similarity. So there's a lot of conversion evolution that can occur where they have evolved, but they're not the same in sequence. So we still tend to find that we have these kind of species that we see as a construct of, excuse me, sorry, my voice is going. Okay, so uh just checking. Okay, yeah. So also useful is we found this useful for, we saw this happen a lot in with SARS-CoV-2, for example, where we get these immune evasive mutations like the R346T. It started popping up in all sorts of lineages independently. And we knew that because we had the whole sequence of these lineages, we could see these multiple mutations and then that would show up. But I do want to just point out to you what would happen if you had two lineages that were very similar who independently got the same mutation? Would we even know that they're different? We wouldn't know. We'd sort of say that's the same one. So this is what I want to point out, that you have to watch out for these kinds of things, and be careful about conversion evolution. So it's useful to, just before we go into this interactive session, I'll just mention that it's good if you can have a device or something that you can use for Slido. I just tend to put my phone next to me and I use Slido on my phone. So I'll just show you that in a bit. But basically, what's useful is to identify orthologs, paralogs, and xenologs. So whether it's homologous or not is often determined by some sort of threshold of similarity, but orthologs are of primary interest because they tend to be produced by speciation, so they tend to have similar function. And we've been sort of referring a lot to the same gene. I mean, that's what we're tending to refer to. And I know some of this will be, for some of you, this might be a review, but I really want to step back and remind you about this fact that you really are trying to find orthologs. The gene tree basically should match the species tree. You can do a reciprocal best blast analysis where you take a genome and you find the sort of the top hit from that sort of gene A in that genome. You see what the top hit is in another genome, and you see if reciprocally that comes back. And that's because many of the sort of paralogous gene families or gene duplications occurred anciently. And so you're sort of trying to find the closest hit. But that can be misleading, and I'll talk about why that can be misleading later. But main point is then you've got paralogs, which are homologs produced by gene duplication. They tend to have differing functions because it doesn't really make sense to have the same gene be duplicated and then doing exact same function. It takes energy to make DNA to replicate things. Usually there will be some sort of divergence of these that are being maintained. Um, for example, you might have a duplication of a transport protein that then diverges to being able to transport slightly different amino acid than the amino acid it was originally designed to transport. So, common way to infer that is you get multiple copies, of course, in the same species, but I'll talk more about how you can do that in a tree. And then xenologs are homologs resulting from horizontal gene transfer. and they tend to adapt some sort of novel adaptive functionality to the recipient organism. So they're often of high interest when you're looking at an outbreak. You want to sort of see, if you have a sudden cluster of horizontally acquired genes in that genome, you want to know, is that like something that's giving it a new um, functionality that might be something that might make this more of a concern, this particular outbreak isolate that's spreading. So, for example, it might have resistance to a drug, or in one case, we saw an outbreak involving Streptococcus pneumoniae, and it was like had a new insertion of some genes that implied capsule change, and that was sort of interest, and there was sort of a concern that maybe there should be more stronger vaccination um, campaign in that region where there was this outbreak because there was this like, novel kind of capsule function that had been obtained. Um, so again, the gene tree wouldn't match the species tree where the rest of the gene tree does match. And or you identify a novel genomic insertion, phase, genomic island, sequence composition differences. And you can detect there too, but I'll talk more about that in a sec. Okay, but what I want you to appreciate is that remember there's in paralogs and out paralogs. Often what we're really do is watch out for these paralogs that you're trying to find things that are the same function. So you want to sort of detect these paralogs. In paralogs are duplication after species divergence. So for example, if you have a duplication here, um, and then a speciation and then a duplication here. This would be in paralogs because they occurred more recently, and out paralogs occurred before species divergence. And it's just always relative to whatever you're looking at. It depends if you're looking at species divergence of mammals or species divergence of human and mice, or are you looking at bacteria strains, or are you looking at bacterial species or genera? And so for example, in this tree, this is useful because, um, M2 and H2 might likely have the same function because they sort of just diverged, seems due to speciation. Whereas in this case, H1 and H3, it's really hard to say what their function is versus M1 if M1 has been studied well. You don't know for sure if both H1 and H3, have sub functionality of this, or if H1 has retained the same function as M1, or H3 has attained the same function as M1, and the other diverged to a different function. So just remember that you've got these kind of in paralogs and out, and you've got orthologs like these. You've also got within species out paralogs and between species out paralogs. So what this means is that you can end up with these kind of different scenarios, and you've got to watch out for this. There was something that was coming up earlier. We were talking about like the same species and doing like pangenome analyses, and you can sort of like use these cutoffs of saying that genes are like, for example, the human genome is, got 90%, 99% the same genes, but it depends how you're defining the same gene. What if you're defining it based on sequence similarity and there's just a bunch of paralogs in there that are not actually the same gene? So like not they're basically very sequence similar. So just be aware that in short, you do need to pay attention to this if you're wanting to sort of find things that are the same versus not in terms of function. An in paralog example, I gave the out paralog of like a bacterial transporter genes that evolved before the divergence of many species. And note that there's been selective gene loss in different species that can complicate analyses. So you can get these situations where where two genes might seem to have the same, like if you have a duplication here and you have gene loss of one from one species and from another species. Actually, I got to come up, I'll show you a different picture later. You can have you can basically have it look like the top blast hit can basically be not an ortholog. It might be that the true ortholog was lost and you're basically identifying a paralog. In paralog example is a recent duplication of some Neisseria opacity genes in a lineage as part of its evolving immune evasion. They're known for that. So you can get these kinds of duplications that are more recent, that are in paralogs. And again, that's, part of it evolving. They're different. Those genes are different. They're evolving, and it's as part of sequence changes to evade the immune system. Um, so I've sort of tried to go on a bit about this, but what I would like you to do is sort of get familiar with some interpretation of trees, of identifying orthologs and paralogs, because again, it's about really trying to make sure you find the right thing. To give you a real world example of why this matters, there was a researcher in my department that was studying a particular gene and found what they thought was the ortholog of that gene, or they found the most similar copy of that gene. It turned out not to be the ortholog, and they were studying that gene for years and then discovered later it was a paralog that had a different function. So, you really do want to pay attention to this. For paralogous relationships, just remember, um, again, the way that can look is you might have two paralogs, and then you would see if they're ancient, you would literally see just the orthologs of species A, B, and C showing up as normal, as, this is one paralog, and this is another paralog, and then you see that that's diverged like an ortholog, like the species divergence here. So let's look at these trees as an example. What are the hollow circles representing? So you can look at this for a second and see what you think. Feel free to add a comment in the chat, but main point is, uh, you see here this duplication here, right? There seems to be, sorry, I should clarify that A, B, and C are different species, and these numbers might be some sort of name for a gene. Um, so here the A1 and A3, this is the same species and same, but two copies of the gene, so that's likely a gene duplication event. Similar here, this is likely a gene duplication event because you've got the gene duplication that occurred before divergence of species A and B, and so you'll get these sort of A and B both have a copy of this, copy 2 of this gene, if you want to call it that, and then here's copy 4 of this gene, if you want to call it that. It's arbitrary. And here is the two copies in those species. So what I want you to appreciate is that these gene duplications, you can either see multiple copies like this, or you can see multiple copies where they still match the orthologous sort of species phylogeny after that gene duplication. Okay, so these are most likely gene duplications. Now, what we're going to do is, oh, I guess this is one more example. So here's another gene duplication again. You'd see A, B, and C showing up. Here's another example where the gene duplication, occurred more recently. So you'd get A there and B and maybe this. And here this gets tricky, right? Like what's going on? This is a longer branch length. Was there actually a gene in A and B here, but they got lost? You've got to watch out for this kind of gene loss, gene gain, and you can get something like a duplication, and then these bunch of species have this gene, but then they get selective loss if they don't need it. So just be aware that that can complicate analyses. So, but really, I can't emphasize enough we're always estimating or inferring evolutionary relationships. And my final comments before we get this interactive piece is that phylogenetic inference allows us to estimate or infer evolutionary relationships. They're often aiding function prediction if you know how to interpret the trees. Data quality is paramount. Having that good alignment, having those good sequences to start with makes a big difference. We've covered some basic terminology and methods and interpretation, but just I want you to appreciate more complicated and efficient methods are needed to deal with genomic data. I've tended to talk more about genes here. Um, but don't discount the value of smaller scale analyses of sets of genes, as I'll show you. Um, but a reminder that phylogeny is best interpreted where you have this well-organized contextual data, geography, patient information, some gene function, where did you isolate this sample, that kind of thing. But we'll go through some examples. Okay, so